Hello everyone, welcome to my YouTube channel. So let's discuss about integrated gene parsing as a topic. So integrated gene parsing is nothing but you are going to predict. For predicting you need few genes because you need to identify the functions of them, whether they're being expressed or not, those kind of stuff. Sorry. So integrated gene parsing, so here you can call it as a gene identification. Okay, it has got two phases: coding region prediction, gene parsing. So coding region prediction, we all know that codon, non-codon, exons, introns and all, most of the stuff it will be there. Whereas gene parsing specifically depends on the determining the gene structure based on the structure. So coding region prediction, so here basically if we speak about coding region prediction, as we know already, it separate modules are built, separate the information data sets are built. That is what they call it as modules in terms of tool, the bioinformatics language. So separate modules are built based on the gene signals, okay, based on the character. That is character, here it is gene signal, to capture the features. Features means again the character of individual functional elements of gene. So what are the different functional elements of the genes? To capture that, the separate features will be there. Okay, they are made as a set of data that is called as modules. So these are then combined to form a gene model. Once you combine all these modules, okay, you will get a gene model that is based on the coding region prediction. So major modules are designed for promoters, exons, introns, and intergenic regions, splice sites, start sites. For every single one of these functionalities, okay, separate set of major modules can be prepared. When you speak about gene parsing, it has got several approaches. Common approach is that you have to consider a set of candidates. So here, why we when you speak about the gene functions, we need to speak about coding regions. Where the coding regions will be there? In exons. So to consider a set of candidate exons weighed by some statistical parameters and then construct the optimal gene. Here when we say some statistical parameters, here it can be of any parameters. For example, for substitution matrices we had Blossom and Pam, you remember. Similarly we will be having in different statistical parameters. So based on that, some parameters will be taken and then construct the optimal gene, like how we used to construct the tree. So now because it totally depends on the structure. So the gene is defined as a constant chain of exons using dynamic programming. So when you speak about DP, that is dynamic programming, so you have to because computer doesn't, the algorithm doesn't know what is gene and all, right? So now the gene, whatever the gene you get, that you have to define it in terms of a constant chain of exons. Now why exons? Because we need only coding regions. Now the neural network, it's one of the highly complicated network in deep learning and machine learning, that is applied either for scoring individual exons, because huge number of data will be there. For that, the best fit model is neural networking. Also it will be applied for either for the scoring individual exons, either for if you are going for scoring individual exons, you have to go for the neural network, or you can go in combination with DP, that is deep learning, or dynamic programming procedure. So that the multiple rounds of networking training will be there, because many things will be considered and combined together, and then the optimal genes are constructed. Okay, so you can say that these are the proper genes. So that is called, it is gene parsing. Okay, so now the integrated gene parsing is nothing but, so these programs, whatever the programs we have been using, they begin by searching signals and performing the coding region analysis. Then again they will go for the homology as well, and once they got all the information, then they will get the optimized results, or you can say optimizing some scoring functions. So then they attempt define exons, and then they will give one or more tentative gene structures. Finally, they will give one or more two tentative gene structures that seems to be, or you can say that is like a most consistent with all the data whatever you are having in your hand. That means whatever the data available, based on that that will represent this is the best what you call optimal gene. Okay, so that is why it is called as integrated gene parsing, because you are optimizing based on all the data which is available at that particular time. Next, we are having, with respect to the continuation of the topic, the second small topic, that is web based tools are there. GenScan is there, that ORF prediction we have discussed about GenScan. GRAIL, codon bias detection, that we will be discussing in another major topic. Under that the GRAIL will come, I mean the codon bias detection as a separate topic will come. The same answer you can write it for GRAIL. And gene finder. Okay, so gene finder usually it performs the discriminant analysis based on the pattern recognition, offers various programs. Many programs are there, like predicting functional sites such as SPL, TSSG, TSSW, FEX is there, N site is there. Next, we are having predicting gene structures. If you are talking about, we are having F gene, F gene-ish, F gene. So these things are there. Next topic, this restriction mapping we will discuss in the end. Before that, I guess we finished one of the major topic, that is any subtopics, predictive methods using nucleotide. Yeah, this we have discussed, detecting functional promoters, transcription, all these things. Whereas gene parsing, finding RNA genes, like web based gene tools, all these things we have finished. Now the prediction methods using the protein sequences, that we will try to finish in the next big video. And then after that, these all things, plasmid mapping, primer design, and in silico, this thing and all. So this will take restriction mapping will take one video, and then rest all will might take one or two videos. So I'll try to finish it off.